The overall goal of this procedure is to study the effects of environmental manipulations, such as chronic food restriction, on relapse to heroin seeking in laboratory rats under withdrawal conditions. This is achieved by first surgically inserting an intravenous catheter into the rat's jugular vein to allow for heroin self-administration. Following a recovery period, rats are trained to self-administer heroin in operant conditioning chambers for a period of 10 days. The next step of the procedure is the abstinence phase, where rats are transported to the animal facility and subjected to a period of restricted or unrestricted access to food. Finally, the rats are returned to the operant conditioning chambers to undergo a heroin-seeking test, where responses on the heroin-paired lever result in activation of a cue light and tone in absence of heroin delivery. Ultimately, chronically food-restricted rats with a history of heroin self-administration will exhibit a robust increase in heroin seeking under withdrawal conditions resulting from re-exposure to the drug-taking environment and drug-associated cues.
The main advantage of this technique over existing methods such as the reinstatement procedure is the absence of explicit operant extinction, which is not observed in the human condition. Furthermore, the use of chronic versus acute manipulations allows for greater construct validity with respect to the human condition.
This method can help answer key questions in the drug addiction field, such as the common neurological mechanisms underlying drug and food-seeking behaviors.